I think it's good to search for and identify individuals that, due to their real tangible contributions in your field and maybe tangential to your field, that can serve as role models. It's good to have role models. And the reason for that is that you want to see people that you respect and you want to learn from. And the main thing is to get inspiration. And it's those kind of people that really push you to achieve great things, and to get on with pursuing your own interests. You're not supposed to follow like a monkey after people, but what you want to do is get the inspiration, see how they do things, and how they push things around and move things forward. And I think the next two speakers that we have, Professor Landegren and Dr. Ulrika Wartman-Berglund here, are really excellent candidates as role models for postdocs. And I just want to put this into a little quick perspective because I don't want to take time away from the speakers. But one of the unique aspects of PULSE, of course, is that we have two tracks. We have the academic track and the entrepreneurial track. And it's really a pleasure to see sort of the interaction between those two tracks. And I think that's one of the things that I think bringing everybody together and treating you as one group really is really important. And I come from a background of academic research, and I've never been worried about inspiration, because if you're pursuing your own questions and going forward in areas where no one else has been, it's inspiring. It's on its own. And so some of the conversations we had yesterday of being bored and finding other places, it's just never come up in my life. All right? And it's always this inspiration and enjoy the open-ended pursuit of new knowledge, right? I've never really thought too much about exploiting the knowledge that I create. But it's my experience now as the director of Campus Solna at the SciLifeLab that really enlightened me about the importance of actually moving knowledge into practical applications that really benefit humankind. And so that's a challenge I think that we all have. And it becomes more and more important as time goes on. But I think one of the aspects that really has brought something forward for me as Campus Solna is that it actually brings engineers, medical scientists, and natural scientists together under one roof. And it's powerful and a magical mix of people, and that's what this group represents. The engineering sciences is a powerful stick that prods things forward with energy. David Baltimore, he's a Nobel Prize winner. He's one of my role models. He's a Nobel Prize winner. He obtained it for discovery of reverse transcriptase. So, a very important finding. And he, in the last year or so, he was reflecting on what was important in pushing America or U.S. science at the forefront of molecular biology. And he came to the conclusion that essentially it was Caltech and MIT, two engineering schools, that were actually pushing the envelope in molecular biosciences to the point where that put America in a dominating position. And again, it just underlies the importance of transferring knowledge into practical innovations. So again, there's two speakers this afternoon really exemplify how this can be done. And so the first speaker is Professor Ulf Landegren here. He's a senior professor in molecular medicine here in Uppsala. He obtained his PhD in 1984. It was 4 or 5 years before I got my PhD, and I actually got to know about Ulf's work as a student. I was taking a course at Cold Spring Harbor Laboratories, and at that time, Leroy Hood, which is the postdoctoral mentor for Ulf, from Caltech, had worked out and figured out how to do capillary electrophoresis to actually automate sequencing so that you could actually go forward and sequence the human genome. And in 1985, that was proposed. We were going to go and sequence all of management. Of course, many years later and everything else, but that was an exciting time. And at the same time, I hope that I'm not taking too much for this, but Ulf was in an amazing environment there, building on his own research that actually has been what I think he's been following up on. And he has a long history of developing molecular tools relying on the amazing properties of nucleic acids to meet analytic challenges in medical sciences, where you basically exploit the principles of proximity ligation to assay single nucleotide changes, analyzing interacting protein, molecules and solutions in vitro and situ, define binding protein complex, and insanely sensitive assays for looking at biological active components in solutions. Broadly significant work. Not surprisingly, Professor Landegren has authored well over 200 peer-reviewed publications and over 50 patents, if I understand it correctly. And he has obviously, many of his papers, if you look at his CV, they garner not just hundreds of citations, there's thousands of citations on each of these papers. It's very impressive. It's impactful science we're looking at here. So the technologies from his lab have been licensed to many leading national biotech companies, and he's given rise to 10 spin-out companies here in Sweden and abroad. And you've probably heard of Olink, and I can list all of these, but it's just really clearly impressive. So it's a real pleasure that Professor Landegren is here, and very appropriate for this community to actually listen to these type of things. And I urge you really to pay attention now, because we're about to witness a true leader in molecular life sciences. So his seminar here now is, or the talk is, Tools for Molecular Medicine. Thank you, Ulf.
Thank you. I feel embarrassed, and I have a hard time to live up to what you said, but I'll do my best. But my first reflection is that I'm kicking myself for not putting in an application for PULSE postdocs. I would have wanted you all in my lab. But anyway, I'll give you a little account of things that we're working on, but I'll start with some propaganda about Sweden and biotech and so forth. Or in general what you're doing. I understand some of you have a more industrial focus and others are more pure academia. But I think either way, I think you probably have to somehow live in both worlds. So I see universities as platforms to build companies. I mean, we should do many things. We're supposed to safeguard knowledge and be service experts in many topics, and train, of course, next generation teachers or doctors or scientists. But one of the features of university is that it's an excellent place to explore things that may or may not turn out to be useful commercially. And maybe we've done a little too little on it. There used to be a feeling that academia needed to be pure and mustn't be influenced by external forces, mustn't be diverted from the goal of finding knowledge. For knowledge's sake. Lee Hood, my postdoctoral supervisor, was a victim of this. He started, even for the US, he was early in starting companies from an academic environment. He was at Caltech, and he was called up to the president and told that it's really not appropriate what you're doing. It's dirty to get involved with money-making business. You should do pure science. And Lee, in his talks now, says that since then Caltech have come around 180 degrees. Now it's the other way around. Now it's almost obligatory. And I think we may overshoot. It may be seen as too important. But when there is an opportunity to take advantage of something you find academically, then I think it's a mistake if you can't bring it further into something that can be more broadly useful. Anyway, so I wanted to show you this Global Innovation Index, which is published every year, and Switzerland has permanently stayed on top. They're very active in innovation, meaning how much venture capital they raise, how many new companies, how many IPOs, things like that. Sweden typically occupies position number 2. In 2022, the US beat us, but things got back to normal. I looked up the situation, and as you see in the top right, for 2025, the situation is the same. Switzerland top, Sweden 2, US number 3. So we're doing something right, and there is an old tradition on trying to work closely between academia and industry in Sweden, more so, I think, than in many other European countries. And I think part of the explanation is the teacher's exemption that I think or teacher's privilege. I think you know about that. So you have greater freedom to take advantage of things that you find, and you don't have to carry around a big burden of ownership by the university, which there's something referred to as a pathological ownership. If you have ownership of people who own your company that are not actively pursuing it, it's seen as unhealthy. Pathological cap table, and if your university owns what you're doing, then they obviously will be rather passive owners. So anyway, Sweden has advantages. I have a few other slides on propaganda. We often complain that there's not enough money to start companies, but actually Sweden mobilizes the most venture capital per capita in Europe. So we shouldn't complain so much. I think we want more, but still we're doing quite well. Another number is the numbers of unicorns, companies worth $1 billion when they go public or sold. And Sweden, there we're number 4 in the world. Israel dominates, but I think it's a very flattering position to be number 4. We beat Switzerland in this case. But just barely. There is also, you can see the yellow part of the segment says how many of the companies stay in Sweden. So Israel creates lots of companies and they go to the US, I think most of them. Sweden have been able to keep our companies, not so much by Swedish ownership, but at least physically, geographically they stay in Sweden, which is a good thing. Please interrupt if there's something you feel I should say or you want to comment on. I also wanted to show our old heroes here, local heroes. This is T. Svedberg and his student Arne Tiselius, both of whom received Nobel Prizes, ultracentrifugation and protein electrophoresis. If you exit the old railroad station, you see a sculpture of this man in the nude. It was very controversial. A friend of Bror Hjort, his friend, an artist, made this rendition of him, and it took a long time before it was publicly accepted, but I think it's quite great. Also, if you go to the University Hall, another controversial thing in Uppsala, there's a saying, Thomas Torrild was a poet and thinker in the 18th century in Sweden. He had a saying which is now a motto for the university. If you go to the University Hall, it says, "Thinking freely is great, but thinking correctly is greater still." So, is that appropriate? What is thinking correctly? But I just want to show you that a colleague of mine, Gunnar Ingelman, he proposed changing this a little bit. So thinking freely is great, but measuring correctly is... and Uppsala is very much about measuring. Now Celsius and Ångström and a couple of physics Nobel Prizes by the Siegbahns, father and son, and the Svedbergs measuring the size of protein, Svedberg and his student. So we've been very focused on technology development here. So turning more into my own travels through science, I was a postdoc, as you heard, at Caltech in the 80s. I came with a background in cellular immunology. I had never seen a DNA molecule. I don't know if I've seen them since, but I had no experience of working with DNA, and it took me quite a long time to get started. My first 3 years as a postdoc, I achieved exactly nothing. I worked extremely hard and tried lots of things, and everything collapsed. But eventually things picked up. But during this time, this was the mid-80s, one of the interesting developments was there was this technique that you all know very well, 2 primers that initiate polymerization and duplicate DNA in an exponential fashion. It's kind of difficult, I think, to see how revolutionary this was. It was not at all how molecular genetics was done. When you did molecular genetics at the time, you would add an enzyme, and then you'd ethanol precipitate and phenol extract, and then you'd dilute it and add the next thing. It was all very laborious and lots of losses. And this, you just had enzymes keeping working inside the same tube over and over, more like things happen in real life. So anyway, I came up with an idea which is much less powerful, which is to have the 2 oligos sit on the same oligostrand and now see, can they be joined by ligation. Turns out that ligases, I came at this for a completely different reason, but I found that ligases have opinions about what is a good match. So if there's a single nucleotide mismatch, they won't ligate as efficiently. So we had a way of distinguishing single nucleotide variation, and most of the variation in the human genome, in the human population, is single nucleotide variation. So it was an early genotyping technique, which we patented and commercialized, was sold to Applied Biosystem since Caltech owned the idea, and commercialized and became a product very quickly. So I was impressed with this speed, and when I got back to Sweden, I started thinking that I really should hook up with a company to work closely with. Back in Sweden, we modified this idea very simply by just having the two oligos to be joined be the two ends of the same molecule. So if the two ends sit down next to each other, then you can join them into a circle. Again, I came to this idea for a completely different and probably incorrect reason. But once we did it, it turned out to open a number of interesting possibilities that we were not aware of initially. One thing is that since this is base paired, it will be helically wound. So this molecule will be wound around its target. Therefore, it's you can't just denature it and have it come off. It will stick in place, and that's why we call it padlock probes. They lock in place. But another thing, like PCR, you have two hybridization events, but they're part of the same molecule. So if you start adding, I think the world record now is 220,000 of these probes, then all you ask is that the 3' end of one will ligate to the 5' end of itself. If you did that with PCR, you had 220,000 PCR primers. You would just amplify the whole genome. So the fact that it's an intramolecular reaction is very helpful. And then there's something called the rolling circle mechanism, which I'll tell you, which turned out also to be very beneficial. Anyway, but we also came up with the idea that this ligation, maybe we could use that for protein detection. So if we put oligos on antibodies, and the antibody would bind the same target protein, then the oligos would be brought in proximity and we could join them. And that gives us a DNA receipt that, yes, we have detected the protein. And later we combine sort of these ideas so that we have antibodies that would, when they bind in proximity, would give rise to circular molecules. And circular molecules have advantages, for instance, for this rolling circle mechanism I mentioned. Are you all familiar with rolling circle amplification? Do you know what I mean by that? No. So it's a common way for virus to replicate circular viral genomes. You just have a polymerase and it will round around the circle. And since there's no beginning and end, it just keeps going. Particularly polymerases like 529 is very good at replicating circular DNA strands. And then you get a local amplification. You don't get a billion small fragments like with PCR, but you get one local amplification, which is very helpful for detection purposes. Relatively recently, we came up with an idea, which I'll describe more in detail later, but which is to do a rolling circle replication, but then interrogate the amplification product with another generation of padlock probes, and then replicate them in turn. So you get two generations. This is something we call super rolling circle amplification, and it turns out to provide some interesting advantages. So, this is sort of the toolbox, things that we have built from, and whenever there is a new need for a solution for a biomedical problem, then we look in the toolbox. This is something we could use that we know how to do that would address this. And over the years, we've built companies that the initial was the genotyping using the OLA, the oligoligation assay, later with padlock probes. We had a company around that. We worked on solid support, multiplex genotyping, spatial functional proteomics, plasma proteomics, targeted genome sequencing, and a company that I won't mention more, but lived for a few years here in Uppsala and then was sold to Agilent. We have technology for antibiotic susceptibility testing, spatial transcriptomics, spatial functional proteomics, plasma proteomics, DNA liquid biopsy, point of care infection diagnostics, and dry blood sampling. So this is companies working on these things, and I ring the companies that I will say something about. But over the years, we've built a number of companies using either the proximate ligation assay or the padlock assay typically. So most things have revolved around the same thing. SampleFacts is a little different. Anyhow, I've been very eager to build a cluster locally, because I think it's very helpful for the companies to work closely with academia, who is the customer for many of these things, since we're building research tools, and be able to go to lectures and be part of that. And also be close to my lab and close to each other. So this is a map of Uppsala. We're now somewhere up above the ceiling, but this is a biomedical center in Uppsala where I work. This is a SciLifeLab building. My corridor is here, or part of this corridor is mine. We have a company called SampleFacts located there, a company called Redley located there. Olink used to be here, but became too big, so they have moved up and have now a nice house downtown. Now they have been replaced by Navinci, and Q-linea is next door, and Rarity is in this corner. But the companies, if they're successful, keep growing out of their premises, so many of them are now on the move. So this is sort of a background of what we do. I thought I'd now go into describing more in detail things we work on, but I realize that not everything will be relevant to all of you, but so I keep it relatively superficial. But as I said, we have this toolbox, and we have started companies around things, and I need to disclose that I am commercially involved with the company, so I have a commercial interest in persuading you that these are fantastic techniques. But I'll say a little bit about plasma proteomics. I'll talk about antibody reactivity, which I think is something we haven't measured enough so far. Large-scale blood sampling I'll talk about. Rare event detection, something I will explain what I mean by that. And spatial functional proteomics. So those are things I will try to cover, but I'll go through it relatively quickly. So first, Olink. I think many of you know the company Olink Proteomics. It's now a company of some around 1,000 employees or so, maybe a little less, 850 or so. And they have a technology which is to attach oligos to antibodies and not ligate them, but do an extension reaction. We had already sold the idea for the ligation to a company, so we just switched to this extension instead. And I just mentioned that in 2005 we sold the right to detect protein via this ligation reaction for 100 million crowns to a US company which is now part of Thermo Fisher. 20 years later, after the patent had expired, we sold essentially the same idea to the same company for 300 times more money. So patents aren't everything. But anyway, Olink now is producing high throughput proteomics, and they are now getting lots of competitors. The patent is out, and many companies are now setting up similar method. Alamar and Spear are two of them, but there are many more. But they have so far been able to maintain a lead together with another company called SomaLogic, a US company, which I have nothing to do with. But I just put here, there are 3 papers that appeared a couple of years ago in Nature full-length papers describing analysis of 50,000 individuals from the UK Biobank with respect to 3,000 protein. Right now, this is now referred to as a pilot study, and now the full-length study, full size, which is 600,000 samples with respect to 5,400 protein. But even this pilot study has given rise to hundreds of papers that from authors who have not done any protein measurement. They don't have any patient samples. They just look through the data, and they've found some interesting things. This is one paper that came out some time ago. From the 3,000 protein, they just picked 200 that varied over life. Some go down the older you get, some go up the older you get, and they created a little algorithm. And here you can see that this was replicated. So it was done in England. Here it was replicated in the Finnish biobank, and it correlated very nicely. If you're 20 years old, you have protein like a 20-year-old. If you're 8 years old, you have protein like an 8-year-old. The interesting thing is that you see that there are some outliers here. People who go around with proteins as if they were much older than they are. Some are a little younger. But if you look at the individuals that have the 10% oldest protein for their age, well, then they have a roughly 5-fold higher risk of dying, 4-fold higher risk of cardiovascular disease. So old protein is a very bad sign. Correlates with ill health in many ways. And this is another study from the same UK biobank study, just showing how different proteins correlate, some extremely significantly, like p-values minus 300, 10 to the minus 300. So with infection, neoplasm, hematological diseases and so forth, often there are proteins. And I just put one slide in from a recent paper from Mattias Ullen's lab in Stockholm, which I enjoyed very much, a science paper a couple of weeks ago, with a lot of information about how protein pattern correlates with disease. So it's sort of disease atlas, which I think will allow us to really tell our health and what diseases we may be about to develop. Based on this, but I just put this slide. So this was there's a Swedish biobank called Scapis. Some individuals were sampled repeatedly over a span of 2 years, and they measured some 3,000 proteins. And this is a mapping showing that people differ a lot in their protein composition. This is looking at all 3,000 protein. But if you look at this, they are swarms. Here's 4 samples of the same color. Here's another 4 of the same color, and so forth. These are individuals. They're colored by the individual. So everybody is different, but everybody's very similar to themselves. So we really shouldn't be comparing, saying you're a 50-year-old woman, you should have proteins like this. You should compare people to themselves. And I'll get back to that. But one of the things that you may want to measure in protein that we know is highly significant for disease is antibodies. And we just inverted the PA assay, so we put oligos on antigen instead. And if there are antibodies, they will bring antigen in proximity, and then you get the same reaction. And we can show that these are systemic lupus erythematosus patients, and controls and patients, and even when you were down to 1 in a million dilution, so 1 millionth of a microliter in the assay, we can still tell who has SLE because of the antibodies reactivity to Rho SSA. And we're currently setting this up for sequencing readout so we can have the same throughput that Olink does. And antibody patterns, of course, tell you a lot what you may be autoimmune against, but also how your vaccine protection is, what infectious disease you have. So I think the world needs a technique like this. This is something we are just in the phase where we're debating. Is it good enough to start a company around, or is it just something we should publish and be done with it? Please.
I was just curious about it's super nice work. I'm very familiar with it. I was just wondering, when you look at the proteins in the previous slides, you said that you compare to yourself but not to others. And I mean, I guess that's in the clinic. When they do clinical work, they have the standard deviation with normality. Have you kind of correlated what you see in your sensitive assays to the variation expected in a clinical lab for some of these?
Yeah. Maybe there are several questions here. So one aspect is what is the precision of the measurement? And this is Olink's work, and I'm not a big user of Olink, so this was Mattias Ullen's work. But it so happens that the Olink assay is not so very precise, unfortunately. Somologic, competitor, has higher precision. I think Alamar also does. Statistically, it's great over many individuals, but you lack in precision. However, a lot of the variation is biological variation, not just technical variation. And this, of course, you can account for if you can take repeated measurements of the same individual. And I think that's what the technology opens. It makes it so much easier. And I think we will see a tremendous price erosion. It will get much cheaper to measure protein. Maybe this is a bigger discussion now. I don't think I answered right. Anyway, yeah, so we're measuring antibodies. This is a population of SLE patients with blood donors, controls, and patients who have reactivity. Some don't have reactivity, some have reactivity. That's positive controls and so forth. So we can do this now in a high throughput. So the blood, again, tells us a lot of our health, and it's very accessible. So we developed something. I should have brought it in. I have it on the in my bag on the other side of the corridor. We have developed a very simple way of sampling, which I think it's different from the rest, which is molecular genetic work. But, so this is just a simple construction. Several years ago, I had a very talented PhD student who unfortunately left my lab and joined Olink. I would have wanted him for myself. But he did the experiment of putting drops of blood on a piece of paper, punching out the square millimeter, and then applying the Olink panel. Very straightforward. And so these are duplicate samples of wet blood where we measure 92 proteins, and the duplicates are the same. So it's the precision is not bad, but it's not perfect. It's not diagnostic quality where CV is below 5%. And the dry, same thing, very reproducible. However, you can't directly compare dry to wet because sometimes they deviate a lot. But as long as you compare wet to wet or dry to dry, and it's so much easier to deal with dry samples. So Johan and I developed a little device which looks like this. A little credit card size thing. Rotates a little bit too long while you're washing your hands, and then it will lie down on its back, and you open up this little booklet, and two membranes are pressed against each other. And now you prick your finger, put a drop of blood, all cells stay on the top membrane, all the plasma goes to the bottom, or most of the plasma goes to the bottom. It takes a second or so. You now give it a minute to dry, and then you can send it in. So it's an extremely simple and inexpensive way of collecting high quality samples, where you don't have to worry that sometimes the centrifugation for the plasma was delayed. And so everything happens immediately. And it's possible now to build really large biobanks where we can compare everybody to themselves. Everybody pricks their fingers every day or every month or whatever, or whenever they feel that they need to measure how they're doing. And here are comparisons of two spots of blood measuring 92 proteins, and just showing that everything falls along the diagonal, meaning that we get the same level. So the precision is good, maybe 10 CV of 10% or so. If we do compare wet plasma to dry plasma, then it's much more variable, as I already showed you. And the same thing is true if we look at the cell layer, wet cell or dry cell, it varies a lot. So you should compare dry to dry, but this gives you a way of building very inexpensive, very useful biobanks. Whenever somebody comes down to disease, well, you can go back 1 year, 3 years, and see what did it look like then. Could we have made a diagnosis then? Was there something to tell us? I'll now switch gears and talk about this rare event detection, as I call it. I had the VR Scientific Research Council grant dealing with how to find rare events, because a lot of biology is about finding things that is very small when it begins, and then it may grow into big disease. So if you want to go into rare event detection, you have to be very efficient in detecting, because if you're looking for rare things, you can't afford to miss too many of it. You also have to be extremely selective, because there will be so much that is not what you're looking for, but maybe close to it. So the demands on specificity or selectivity grows as you look for more rare things. If you look for albumin, you can use Comanche blue and you'll find it. But if you want to look for a point mutation, then you really need to input a lot of specificity. And preferably you should get something that cannot be mistaken from a positive reaction. Little dust shouldn't be mistaken for a positive reaction, because you can't afford to have too many false positives. So we came up with this simple idea. And this was another PhD student, later postdoc in the lab, Lei Chen. He incorporated a sequence in a DNA circle, a sequence that may or may not be mutant. For instance, the situation is that you know somebody has a tumor, and you know the tumor has particular point mutations. But the rest of the body, of course, will not have this point mutation. So by drawing blood, you can now see, does any of this blood derive from the tumor? So it's a sign that there's still tumor in the body, or it could be leukemia. So we make circles. Lay tends to make it by just doing a few cycles of PCR and then circularizing, but there are other ways. Then you replicate it, and now you generate a few hundred copies of either wild type or mutant. So if the circle had a mutant, then you'll get copies of the mutant. And then your genotype. You have padlock specific of mutant or wild type, and they all will sit down, and they're pretty good at distinguishing point mutation. However, they will make a mistake every once in a while. I show a mistake here. The blue circle shouldn't be there, but it will happen, and that would set a limit to how sensitive it could be. But since we already made 1,000 copies, we don't care if 1% is off. So then we just replicate the circles that form, and well, we'll replicate the mistaken one also, but it gets sort of diluted out. It's a majority vote mechanism, which means that we're extremely selective. So we can show, I forget what data, yeah, I have some data on that. But it turns out we can find very rare mutations. But also, these are two linked amplifications of the same molecule. The same molecule first gives rise to this guy that hangs on to the starting molecule, and then these guys that hang on to this molecule. So they all form one big cluster of DNA. And you can see a picture of such a cluster. There's the size of a little cell, or a platelet at least, a couple of microns in diameter. So whereas in PCR you get a billion copies of what you're looking for, which is fantastic and helpful in many cases, here you just get one molecule. For every starting molecule you get one huge molecule. So you can now use it to count target molecules. So you circularize the molecule, replicate genotype, and then replicate the second one. And this is a rather complex reaction where you want a circle that's already wound around something. This yellow circle is wound around this blue first generation Rolling circle product, and now we want to replicate that and generate this red strand. I wasn't so sure it would work. In fact, I advised Lay not to do that experiment, and he didn't bother as usual what I said and did it, and it works. So I'm grateful to him for not listening. And this is results. So here we took, these are 4 mutations that are common in acute myeloid leukemia. Isocitrate dehydrogenase 1 or 2, and 2 different mutations in each gene. So we took genomic DNA from cells that had this point mutation and diluted it in genomic DNA from cells that did not have it. And as you can see, when we're down to 1 in 100,000 of the genomes comes from the tumor, well, we can still see a signal above background. We're comparing ourselves to digital PCR, and digital PCR gets down to maybe 1 in 1,000, and then it's just noise. So we can find rarer events. And rare events means maybe earlier detection. I don't think I brought slides on that, but or maybe I did. Let me just check. Yeah, no, I have one example of that, I think. So, but first I'll just show you. This is a study we did with UK UCAN biobank, local biobank on cancer patients, where they had been sampled at the time of diagnosis, and then had their tumor sequenced, so we knew what mutation they had. And then we just looked how much of that mutation could we find in plasma, because cells will leak DNA into plasma. Both malignant cells and normal cells will do that. And we found that some of the individuals had very high levels. These are the frequency with which we found it. And some had very high levels detectable by a simple sequencing run. Others could only be detected by super RCA, and some were below our detection threshold also. So that was the situation at diagnosis. Then the patients were operated on and tested after a little while, and you can see that many of them became undetectable, as one would hope. Some stayed undetectable. Some went from undetectable to detectable, to our surprise and disappointment, and some remained detectable. So there were 6 individuals here that had detectable levels. 2 of them by sequencing and 4 by our method. So you may now ask, what happened in the long run to these patients? And they haven't been followed up so very long, but this was the pattern when we checked it. All the ones that were detectable went on to have a clinical relapse in their colorectal cancer. All the ones that were undetectable remained healthy. So it's highly important information clinically. If you can tell the patient, you're cured, we don't have to worry about you. You probably can't be so sure, but at least it's a very strong indication. So I think this is how healthcare will happen in the future. We will be monitoring patients and trying to get rid of the last remaining sign of a malignancy by changing therapy and see if we can get rid of it. I brought another example which is a little special. It's a very difficult mutation because it's located in 85% GC rich, so PCR doesn't work very well. And the mutation is an extra G in a row of 8 Gs. So you get 9 Gs instead of, very difficult to detect. We're not so, sequencing can get down to 1 in 20 or so, and we only get down to 1 in 2,000 or so, but still. So this patient comes in, lots of mutations in bone marrow, barely detectable by sequencing in blood, although clearly detectable by super-RCA. And then time goes on, the patient has a stem cell transplantation, and levels keep being above or around detection level, and then things start taking off, and the individual has higher and higher levels, blood levels of the mutation, and then eventually at this point there's a clinical relapse, and now it's obvious that the patient has not been cured and needs better therapy. But we had a 3-month head start to see that things were not going well, and the patient would have needed a better therapy. This was all done retrospectively, so we had no way of influencing the health care of this individual. But with this information in hand, the clinician would have been able to adjust the therapy to try to get rid of this remaining malignant clone. I just threw in another. This is a recent study from a lab in Spain, publishing interesting study on a KIT mutation that is relevant for mastocytosis, where they showed that by comparing allele-specific PCR versus super-RCA, we were able to find more, all these individuals who were suspected of having the mutation, 27% of them could still not be detected by super-RCA, others could, but with the allele-specific PCR, 46% of the patient could not be detected. So at least it could be that some of the 27 shouldn't have the mutation also, but so we were more sensitive, or the lab in Spain. Finally, I'll just mention the localized protein detection, so spatial biology, which is very much in focus now, and where we have this localized protein detection. But first I'll just show you one slide. So this is a paper which was published in 1941, Kunz et al., which was the first paper to say that you can take an antibody, put a fluorophore on it, add it to a tissue section, and see where it lands, and you get a fluorescent staining. So immunofluorescence, published 1941. And since then, that's what everybody's doing. Sometimes they're using an enzyme-linked reaction instead of fluorescence-linked reaction, so immunohistochemistry. It's a surprisingly stable assay, although people have, since 1941, people have been complaining about it. Nobody measures protein in solution with a single antibody. Olink needs pairs of antibodies. Everybody uses pairs, sandwich ELISAs, because that way you can ignore some cross-reactivity. In situ, everybody's using this method. So we've been the only ones using pairs of antibodies also for localized detection. And here's one example where we did, it's kind of maybe a strange assay. It's a capillary electrophoresis with separated protein, and then add primary antibodies that are either rabbit or mouse, and then detect them. And as you can see here for aldose reductase, we get a peak where we think it should be for both the rabbit and the mouse. However, the mouse has an almost equally high peak over here, and the rabbit has something over here. For this other protein, it's similar pattern. They agree that there's a peak here, but the mouse sees something here and the rabbit sees something elsewhere. For C34, it's even worse. They can't even agree, the mouse and the rabbit. So if we now do this assay instead, where we require the two antibodies to bind together, then and only then will we get the circle. and we get this nice pristine peak for these. And in this case, we don't get a peak because they can't agree. So one or both of them are wrong about what protein they bind. So binding with dual recognition gives you more specificity. And here's an in situ example. This is a monoclonal antibody. I don't even say what. It's ref1. And as you can see, everything is pretty brown. Some things are browner, but it's not really a good way of saying where is the protein located. The other monoclonal antibody, again, generally brownish. However, when you apply both antibodies, then you get nice staining where the signal should be and not elsewhere. So dual recognition is helpful. And there's some more examples. I think I shouldn't go into that. But you can also use this to look at things like how signaling happens. So we can look for phosphorylations or interactions and follow that over time. And we've also developed an assay to see if PD-1 and PD-L1 are interacting to say that an oncogenic therapy against this immune escape of tumors could be helpful. And that way we were able, or a group here, I wasn't involved in the study, it's Patrik Micke and colleagues who did a study on lung cancer and showed that they could better predict who would respond to therapy by doing this assay. And since then we've now set up multiplex assays. So this is a 9-plex assay where we're following the EGF receptor signaling cascade. And I'll just show you, but it's difficult to evaluate. These are cells that have not seen EGF, and these are cells that have seen, on the right, cells that have seen EGF, and we can show that we can upregulate all these signaling events. Here's phosphorylation and other signaling events. So, and you can do this also in tissue sections. And with this, I think I've come to the end. I think I'm running out of time. So these are individuals who have funded us, and this is the group plus the company SampleFacts is also represented here at my Summers house. So maybe the detail of what we have been doing are not so relevant, but I think the way of working closely between academia and industry could be maybe relevant for things that you will be doing. So with that, I close and take any questions.
Fantastic. We have some questions. Go ahead.
My lab has never worked on microRNA. Other people have used the padlock probes for microRNA detection, and I think they're very suitable for that purpose because they get a lot of information out of a small target sequence. The problem with microRNA is that there's so little to take on. Yeah, but that's a possibility. There are papers on this. I'm not convinced that microRNA are very valuable diagnostic markers. Maybe they are, but I feel people have been working on it for a long time and not so much has come out of it. So I put my money on proteins over microRNA as biomarker. Do blood levels correlate to tissue levels? And it's a very general question that I can't answer. In some cases, it will not correlate at all, I think. You have very local disease processes that hardly manifest elsewhere. In the case of mutations, you have the advantage that they are so distinct. So we can look for these rare mutations to see if even a very, very small fraction of what happens in the tissue comes out into blood. We can find it, maybe. But in many cases, you probably cannot. From a practical point of view, any disease that you can diagnose in the blood is going to be much easier to diagnose. So that's a light post we look under. Blood is so easily accessible. However, of course, it would be ideal if we could get tissue with the same ease, and that's not so easy. Tissue sections. There's a Swedish speciality in fine needle biopsy, which isn't so common internationally, but pioneered at Karolinska, which gives you access to tissue with minimal invasive means, but it's not so popular. I think it should be used more. So I don't think I can answer more specifically. But my main advice is make sure that you work together with people who know what they're doing. Myself, I don't think I would have been able to do any of the company. I've had fantastic students that have been very important and really driving and much more inclined towards industrial applications. But I've also been very careful to have people with industrial experience in everything we've done. But this is a little list of things that you may want to think about if you think about starting companies. But unfortunately, I didn't have time for that. Yeah, so this is referred to as measurable residual disease, MRD, or minimal residual disease. And it's been around for a very long time in hematology. In chronic myeloid leukemia, you have a very characteristic translocation between BCR and ABO, which is very easy to pick up by PCR sensitively. So that's standard practice since 20 years or more. But only now are we getting methods that can look for more complex mutations. So it will take its time to be integrated in clinical care, but I'm convinced it will come there. The situation now we have is that we get more and more possible therapies, for instance in hematology, and clinicians are in the blind. They have to choose some of all the possible therapies, and then they give it, and then they wait, and after 2 years the patient comes down and is still sick or a recurrence of the disease. So by being able to measure if people respond and pick the right, this is not working, we should switch it to a new therapy. I think that's going to be really important, but it will take its time. We have a little company called Rarity that's trying to pursue this.